So chapter 9 is everything that you wanted to know about DNA. It's everything DNA related. So kind of the scoping out the chapter, we're going to talk about how DNA is packaged in chromosomes. We're going to talk about DNA replication, talk about the transcribing of DNA and then translating it into proteins. We're going to talk about mutations, so changes in DNA, and how we can get DNA from one cell to another cell. So lots of DNA in this PowerPoint. So we're going to start with this slide with genetics and genes. So anytime we're talking about DNA, it usually is also talked about with genetics. Genetics is the study of heredity. It's the transmitting traits from a parent to offspring, but the only way you can transmit traits is if you transmit the DNA. And so we're going to talk about all about DNA and how it can get from one cell to another cell. Now, DNA is packaged in chromosomes, and we'll go over a little bit about chromosomes, but it's the DNA that's found in the chromosomes that can be found in the cell that code for various organism structures. But we're going to focus first on the chromosomes and the DNA that's found in those chromosomes. Now, a genome, it's the total of all the DNA in a cell. So you may have heard in the news, they talk about, oh, we've just sequenced the genome for COVID-19, because we just did here in La Crosse. We sequenced the genome for this particular virus. That means we've sequenced all of the DNA, all of the RNA found in that particular virus. We know all of the base sequences. We know all the A's, T's, C's, and G's found in a particular cell or organism. So a genome is all of the DNA. Now, the DNA usually exists in and is found in chromosomes, but it's not the only place that you can find DNA in a cell. Some DNA can be found in non-chromosomal sites. You can find DNA in the mitochondria, if an organism has it. If an organism has a chloroplast, you can find DNA in the chloroplast. And if an organism has a plasmid, it's another place that we can find some DNA. Now, a chromosome where we find this DNA is neatly packaged DNA. Now, eukaryotic cells, their chromosomes are linear. If I had to draw one of your chromosomes, I would draw an X. That's 2 lines. So chromosomes are linear, and there are multiple of them. You have 23 pairs for a total of 46 chromosomes in your cells. Other organisms have chromosomes in the hundreds. And so eukaryotic cells have multiple chromosomes, and they're all linear, and they're all found in the nucleus, because eukaryotic cells have a nucleus. Bacteria and prokaryotic chromosomes, there is no nucleus. Their chromosomes are circular instead of linear, and they only have one single chromosome. And because there is no nucleus, we find that one single circular chromosome in what we call the nucleoid region. It's not nice and neatly organized. It doesn't have an envelope, but it's where we find the DNA, so we say it's kind of nucleish, so we call it nucleoid. So this is showing those comparisons. Eukaryotic cell, you're going to find those linear chromosomes, multiple of them, packaged in the nucleus. In a bacteria or prokaryotic cell, they have one single circular chromosome. And viruses can have either DNA or RNA. It's not packaged in anything. Viruses don't have a lot more than just some protein and some genetic material. But viruses can have DNA, or they could have just RNA as their genetic material. Now, a chromosome, we take all of that DNA and we group it in little tiny genes. So a gene is just one little snippet, one little part of that full DNA sequence, and that little snippet codes for something. It might code for hair color, skin color. It might code if you're a cell that has a flagella. It's got you need some type of genetic material that codes for that flagella. So it codes for something. So it's a site in the chromosome. It provides some type of information for some cell function, whatever it is. And we group all of those little snippets of DNA into structural genes, anything that codes for various structures, genes that code for making RNA, and genes, those little snippets that code for gene expression. They're called regulatory genes. Now, all of the genes together, so all of those little snippets constitute what we say is your genotype. It's all of those genes. What do you have that, what are all the genes that code for all the things that make you, or makes any organism or any single-celled organism them? The phenotype is what do all those genotypes do? So a gene is the snippet of DNA that codes for, say, hair color. So the phenotype is that physical, I always think, the pH for phenotype, pH for physical characteristics. What is that outward physical characteristic? What is your certain hair color? What is your, if you are a single cell, do you have flagella? Do you have cilia? Can you make a specific enzyme or not? So all of those outward traits is the phenotype. Again, all controlled by the genotype, all of your genes. Now, packaging of all of that DNA and all of the genes, viruses, they're tiny. Again, we didn't even know bacteria existed till microscopes, but until we had some of the best electron microscopes, we didn't know viruses existed. Viruses are small, and their genetic material is small as well. The smallest viruses only have 4 to 5 genes. So that's only 4 to 5 things that particular virus can control, different characteristics. An E. coli, a bacteria, can have somewhat, over 4,000 genes. A human cell, or over 31,000. The more advanced, the more genes you have to control all of those advanced traits. Now, when we package the DNA is packaged, like it bundles itself around and around and around. In prokaryotes, we use enzymes to coil up all of that DNA in those single circular chromosomes. In eukaryotes, we also use different types of DNA, just a little more complex because we've got longer DNA strands that we have to get coiled up and packaged inside of each of your microscopic cells. So this is just showing how we can package that. There's your double helix DNA. We're going to package it. We're going to wind it around different proteins. We're going to wind all of those around each other. And then we're going to kind of wind those around each other and so on. Until all of a sudden you have this very large structure of chromosome that has 6 feet of DNA in it. When you think about it, you have 6 feet of DNA. It's so skinny. 6 feet of it can fit into one of your microscopic cells. Now, structure of DNA, well, it's a double helix. And so this might be where you recognize a little bit of these next couple slides. DNA, it's got a deoxyribose sugar, it has a phosphate group, and it has one of 4 different nitrogenous bases. Your adenine, your guanine, your thymine, and your cytosine. Now, the nucleotides bond together, and we have a sugar phosphate backbone on the outside of the DNA structure. So you've got a deoxyribose sugar, a nitrogenous base, and your phosphate. So the backbone is your sugar phosphate, and then your nitrogenous bases go together, they pair up, and they form a hydrogen bond. So you have a double helix. You have 2 strands of these nucleotides, and they're connected together with hydrogen bonds, which are weak bonds. Now, a little refresher, if you didn't remember, adenine pairs with thymine and cytosine pairs with guanine. So I've got it on there what they pair with. Another picture showing a couple other structural. Again, you've got your sugar phosphate backbone on the outside, and you have your nitrogenous bases paired together on the inside. My other note about your structure of DNA is that the 2 strands do run opposite of each other. So they are anti-parallel, means they run opposite of each other. If I notice the arrow, one goes this direction, the other goes the other direction. Now, they call one end 5', that's what the little asterisk is, and the other end is 3'. And so you can see that they are opposite of each other. They're opposite. If this side is the 5' end, the other side is the 3. If this is the 3, the other end is the 5. They do run opposite of each other, which becomes tricky coming up when we get into DNA replication, the fact that they are 2 opposite running strands. So here's my anti-parallel part, showing that if one end is the 5, the other end is the 3. Now, the DNA structure, One, and I'm like, we have to maintain that same base sequence anytime we make new DNA. We can't change up the DNA. That DNA codes for something. But, and I'm like, there is a variety in the different bases that are there so that different organisms are different. And we do need to protect that so that the phenotype of each organism is also gonna, convey what it's supposed to convey. Now, when we make DNA, the strands, each strand, because it's double-stranded, will become the template. It becomes what we're going to use to copy to make a new strand. Now, a little concept check. I'm going to skip through these because they're on there for you. But when we make new strands of DNA, the original strand, we call it the parent DNA molecule. I'm like, we pull it apart. Luckily, those hydrogen bonds that hold the two strands together are weak bonds, so we can pull it apart. And they serve as templates to make brand new strands of DNA. Now, it's called a semi-conservative process. So when we take, I like how they color code, and like the original DNA, double-stranded DNA is blue. Well, we're going to yank those bonds apart, and then that blue becomes the template, that if it had an A, we bring in a T. If it had a C, we bring in a G. And so that original strand is still present. It's just by the end of making new DNA, you now have two strands of DNA. Now, when we make new DNA, where it all starts is called the origin of replication. It's where we're not always going to copy all 6 feet of DNA or however long it is. We usually just want to copy certain areas. So where it starts is the origin of replication. And as soon as where it starts, that DNA has to get yanked apart. So we have what's called replication forks. It's where we're going to pull that DNA apart and start making new DNA. So this is showing that this is an enzyme, and it's pulling the two strands apart. It's ripping apart those hydrogen bonds. Now, we have 2 strands here, the original blue that we're going to copy. But this is, again, is where we run into problems, is those 2 strands run opposite from each other. And the enzymes that are used to make new DNA, that lay down this red, the new bases, the new A's, T's, C's, and G's to match up with the original A's, T's, C's, and G's, on the other strand, they only can add on a 3' end. So when they're making it, as you're unzipping going this direction, that's totally fine for this particular enzyme. As it's unzipping, it just keeps adding the correct base. If this had an A, it would bring in a T. If this had a G, it would bring in a C. And it follows along. But we run into the problem on the other strand. Because it can only go and add onto a 3' end on its new strand, it has to go in reverse. Because the other strand is going the opposite direction. And so it runs into problems. So we still have an enzyme, and it's still going to add bases, but it has to go in reverse. So as we are unzipping going this direction, we're actually making segments going the opposite direction. So to do that, we do it in small little sections. So it kind of jumps ahead and makes a new segment going in reverse. And so I'm like, it will jump ahead, go in reverse. It will jump ahead, go in reverse. And we end up with these little fragments called Okazaki fragments. Now, we can't just have fragments of DNA. We do have other enzymes that come in, other parts to the game, primers, things like that, that will link these different fragments together. So it is still going to be one continuous strand of DNA. But it takes a little longer. So we call that strand the lagging strand. It takes longer to make all of these individual fragments going in reverse. And then bringing in different enzymes, different players to the game, and putting them all together. So this is kind of showing those events that are happening. Now again, it's summing up this, the fact that we are ripping apart the double-stranded DNA. We're using enzymes. This particular enzyme is an enzyme called DNA polymerase. Enzymes, it's an enzyme. DNA polymerase 3. It's one of the biggest enzymes that plays a role because it's the one that brings in the new DNA bases, the A, the T, the C, and the G to match up with what was originally there. So I'm like going through the different steps that, again, we are going to unwind the DNA using an enzyme, yanking it apart, called helicase. Again, we have a lagging strand that runs opposite to the leading strand because of that 5' to 3' end. And we've got different primers that help direct where the DNA polymerase is going to start laying down bases. Now, in the lagging strands, yes, at some point we have to get rid of the primers that start off and then eventually join all of the nucleotides together by another enzyme, enzynase, called ligase. This is showing another picture of making the new DNA on the leading strand. Again, as we unzip going this direction, it's totally fine for the leading strand. And I'm like, that DNA polymerase is going to bring in the correct A's, T's, C's, and G's, no problem whatsoever, adding on to the 3' end of our new strand. The lagging strand has the issues because it can only add on to the backside. So it makes these little fragments. It starts and goes in reverse, will start and go in reverse, those Okazaki fragments. But yes, at some point we're going to have more enzymes, we're going to remove primers, and we're going to link everything together. Now, this video, you should be able to click on it. I'm not going to. And it shows a nice picture of all of that happening. There's kind of a lot of information all there together. So I'm like, you can watch it a couple times. It's a short video, lots of information packed in there. Now, in a prokaryotic DNA, again, it's still going to have all the same basic steps. Just a unique difference, though, is that they don't just have a line, a linear DNA fragment. I'm like, there's everything's wrapped up in that single circular chromosome. And so they're still going to do replication, but they have like 2 forks. They're going to go opposite of each other. It's still double-stranded DNA, but they have 2 forks that are going to go in opposite of each other, making all the new DNA. They're still going to have enzymes like ligases that link everything together. So here are those, they're copying it, they're making that new DNA fragment, and then at some point they have the little nick where they're going to separate those 2 DNA fragments, and now you get 2 chromosomes instead of 1. Now, the DNA code, all of those A's, T's, C's, and G's that are found in DNA, we generally read them 3 base pairs at a time. Each 3 letters code for some type of amino acid, which is used to make proteins. And we do this through a process called transcription and translation. So that DNA does have to code for something. Now, we're going to take that double-stranded DNA, and I'm like, we're not just going to make new DNA. That happens when we want to make new cells. We have to make lots of new DNA to go along with it. But we can take that information that's in the DNA and we can make other things. If we need to make a particular protein, if we have to make a particular enzyme, which is a protein, we need to be able to read the DNA and make from it whatever it is the cell needs. So we have to be able to take that DNA, read it, make a copy of it, and then eventually make a particular protein, chain of amino acids, to do whatever it is we need to do. Now, like I said before, we generally read DNA. Our cells read DNA 3 letters at a time, 3 nucleotides at a time. And every 3 nucleotides is called a triplet, or more commonly called a codon. And it's because each of those 3 letters, those 3 bases, those codons, they all code for one specific amino acid. Let me show you. And I'm like, so if we've got 3 letters, again, we're going to take DNA, we're going to read it 3 letters at a time. There's your one codon, your triplet. And when we transcribe it, we are again transcribing it. We're going to transcribe all of it. We're going to make a copy, but again, keeping it kind of separated as 3 letters at a time. It's because these 3 letters code for this particular amino acid, and these 3 letters code for this particular amino acid. And I'm like, so those 3 letters, those codons, code for specific amino acids. And depending on an amino acid, depends on what protein you make, depends on what you need that protein to do. It's all dependent on shape. Now, RNA, we talked a lot about DNA, but RNA is a big factor in all of this as well. If I go back that we're going to take DNA, and the first thing we're going to do before we can make any protein is first make some RNA from that original DNA. Now, RNA stands for ribonucleic acid instead of deoxyribonucleic acid. A couple differences from RNA and DNA is RNA is single-stranded, and RNA contains one different nucleotide base. Instead of A's, T's, C's, and G's, it has U's, T's, C's, and G's. So it has uracil instead of thymine. And uracil will pair up with A just like T would pair up with A. They're similar enough, they recognize each other as being something they can pair with. Now, the reason why it's called ribonucleic acid instead of deoxyribonucleic acid, there is a different sugar. It's ribose instead of deoxyribose. Now, messenger RNA, that is going to be the first thing that's made when we read DNA. So we transcribe or make a copy of it. I'm like, it's RNA, so it's single-stranded, and it has the bases, the A's, the U's, the C's, and the G's. But again, we read it as 3 letters always at a time. So each 3 letters is considered a codon. Another type of RNA is called transfer RNA. It has a different structure. Messenger RNA is called messenger because it carries a message. Transfer RNA is called transfer. It's going to transfer amino acids. This is the RNA that's going to be able to read the message on messenger RNA and bring in the transfer part, bring in the correct amino acid. Now, how it can read it and bring in the correct one, at the very, very bottom of transfer RNA, there is a 3-letter sequence called an anticodon. And it can read the codons, like GCU, it can read those 3 letters, and it recognizes those 3 letters because it has the counterpart to it. And so if this is the anticodon UAC, it would recognize any codon that has A, because that's what U pairs up with, U, because that's what A pairs up with, and a G. So if it sees an AUG, it knows, I recognize my counterpart, and it will bring in the correct amino acid at the other end. The last kind of RNA, because there are 3 main different types of RNA, is called ribosomal RNA. This particular RNA makes up a large chunk of, based on its name, the ribosome. And it's that ribosome that's going to make proteins. That's the main job of a ribosome, is to make proteins. And it's that transcription, translation part that is where we find the ribosome making different proteins. Now, for transcription, it's the first stage of reading DNA. And there are 3 steps to transcription. There's initiation, elongation, and termination. So we're going to start, we're going to do something, and we're going to stop. Now, transcription, the word it comes from transcribe, which means to make a copy. Now, my best way to try to, simplify what's happening in transcription. So DNA codes for everything that a cell needs. Everything. It has all the genes, thousands of genes that code for anything. But we don't want to copy all of it. We just want to copy one small little gene. Now, my analogy is if you had a book in the library, the public library, that was like the book of life. It had all the information, everything. It could tell you everything in the world that you wanted to know. And there was only one book. They're not going to let you check it out. It has to stay there. Well, that's kind of like DNA in the nucleus. It has everything you need. It's not leaving the nucleus. But instead, if you can't take the book out, you can make a copy of a page. And that's what's happening in transcription. We're making a copy of a page of a book. We're making that just a copy of the information that we need. We don't need the whole book, just like we don't need all the genes. We just need one gene. We just need one page out of the book. So we're going to make a copy of just one part of the DNA. Now, picture-wise, what that can look like, and I got a couple different diagrams. And I'm like, we have that original double-stranded DNA. Well, it has to get separated for us to make the RNA, which is going to be the orange, yellow-orange. on this particular picture. So we first have to separate out the DNA because we're only going to copy one strand. And the cells know which side, which strand to copy. They pull them apart, and we use more enzymes. This is now called RNA polymerase, and it reads the DNA, and it brings in the correct RNA base. If there was an A on your DNA, it would bring in a U because it's making RNA. If there was a T on your DNA, it would bring in an A nucleotide. If there was a G, it would bring in a C, and so on. And it would continue down the line until you get to some type of ending sequence that stops, that we will have now copied our one gene, our one page out of the book, our one snippet of information. I've got a video of this. That the video does both transcription and translation. I recommend watching it. Again, lots of information in this video, and they show how fast this happens because they do it in real time on how fast this happens inside your cells. Now, my note, I'm like, the video goes through both transcription and translation. If you're going to watch it, just watch it up to the transcription part and then stop it, because I've got the same video link coming up to kind of go over the translation step. Now, the translation is you've made your snippet of messenger RNA, you made your copy, your page out of the book. Well, now that's information, but it still doesn't help us yet. It's got our information, but we haven't made anything yet. So now we have translation, which means, we're reading something, we're translating it. And in our case, we're going to translate the message from RNA into another language called amino acids. So we're going to put it into a more readable form. And I'm like, for our needs, we're going to just translate it from messenger RNA into the language of proteins, which is amino acids. Now, it also has several different stages. And because our final outcome is going to be proteins, the ribosome is going to be involved because the ribosome makes proteins. So it occurs in 5 stages, all happening inside the ribosome. The ribosome is this big blue blob. And so we're going to have initiation, elongation, termination. We're going to have the protein folding and processing. Our ultimate, we're going to make a protein. Now, just showing another picture, we're going to take our messenger RNA and we're going to translate it into the language of amino acids. These are 4 different amino acids to make a big chain of amino acids, which is called a polypeptide. And then once that polypeptide is made, it folds and shapes itself into the correct shape of the protein. Now, when we do read, because I'm going to go back a second before I get through the steps of translation, when we go from messenger RNA to the protein, we read everything 3 letters at a time. AUG, CGG. We're reading everything in codons, 3 letters at a time. And the main reason we have to do that is these 3 letters, AUG, that codon codes for a specific amino acid to get brought in, called methionine. These next, the CGG, codes for a specific amino acid called arginine to get brought in. So in your ebook, and I'm like, it has this table. I apologize, I didn't write down what table number it was. But it's this table that it will show that every 3 letters codes for a particular amino acid. These are the codons. Now, how to use the table, because you're going to have to do this for the worksheet this week. The first base in your codon, so whether it's the A, the G, the C, the first base in your 3-letter codon are over here. So if we're going to go back and we're going to find AUG, but we don't want to just look around on the table. There's a lot of different codons on there. So we're going to translate AUG. So our first letter is A. So on the left-hand side of the table, we have our 4 possible choices for the first letter, and our first letter is A. So anything in this row, if you notice, all of it starts with an A. Our second letter is U. So we go across the top, and that's where the second base position is. Ours is U. Again, there are the 4 options. Ours is U. So we know we're in this 3rd row, but we're going to be in this first column. And if you notice, yes, everything in this box from here starts with AU. The last letter on our codon is G. And that's where over on the far right-hand side, in each of these individual rows, it has those 4 options. Ours is G. And so we're in the bottom row in our box. And there's your AUG. Now, if you notice, there's a reason why it says start. It's because this is usually where the ribosome starts translating messenger RNA. It usually tries to find an AUG, and it starts the translation process, and it brings in the amino acid methionine. Now, if you notice, there are 3, 2 here and 1 here, codons that mean stop. It means there is no particular amino acid that matches up with those 3-letter sequences, those particular codons. And so the ribosome, if it ever encounters one of these 3 codons, it stops the translation. It stops bringing in any amino acids, and the protein's done. So, looking through it again, we've got DNA made into messenger RNA. We're reading it 3 letters at a time. The transfer RNA does have the opposite codon called an anticodon, and the transfer RNA will bring in the correct amino acid. In our first case, methionine. Now, all the steps on here, and I'm like, the translation part, I like pictures more than words. So here's my pictures, is that in your ribosome, which does the translation, it brings in that messenger RNA. It feeds it through, and as the ribosome will start to read, again, starting at that first codon, the AUG, it's a start codon. Transfer RNA recognizes that particular 3-letter codon, and it brings in the correct amino acid. Now, as we keep feeding this messenger RNA into the ribosome, we keep reading it 3 letters at a time, and the transfer RNA will come into the ribosome when it recognizes the particular codon, and it brings in the different amino acids. So we've got transfer RNAs, our first 2 coming in. I'm like, they recognize their counterparts, and then they drop off their amino acids, and we link the amino acids together with a bond called a peptide bond. The transfer RNAs leave. They left their amino acid here. We've got our first 2 amino acids, and the messenger RNA will continue pulling it through. More transfer RNA comes in, more amino acids get brought in. We keep linking all of them together. And so it continues until it eventually reaches one of those 3 stop codons. And I'm like, you're going to end up with, in the end, a whole bunch of amino acids, sometimes thousands long, a whole bunch of amino acids that will form a particular protein. This is showing that it will start at a start codon, so that ribosome comes together at a start codon. It links all of your amino acids together. You can see at the top your new amino acid chain, your polypeptide being made. And as soon as it encounters one of the 3 stop codons, and I'm like, it's done. It's like, all right, I'm done. I don't have any more amino acids to get brought in. I made the protein that I'm going to make. So this is just showing you, yes, there are 3 different stop codons, and then the ribosome goes away. Now, what makes it faster is, cells can do this very, very quickly, but to speed it up, because sometimes you need lots and lots of proteins, you can have multiple ribosomes reading a messenger RNA sequence at the exact same time. So if this is the messenger RNA sequence, this yellow strand here, you can have multiple ribosomes reading it. Now I'm like, this is showing an actual picture of a messenger RNA, multiple ribosomes attached onto it, reading it, and you can see those little RNA, or the little amino acid sequences that we're making coming off of it. So it speeds it up. You can take one book page and make a whole lot, mass produce a whole lot of protein, whatever it is that we're going to make. Now, here's the video if you didn't watch the whole thing. You again can watch the whole thing of going through what's happening in transcription and in translation. This is just showing that basic steps of transcription and translation is going to happen in both prokaryotes and eukaryotes. Where they happen can be different. Again, a eukaryotic cell does have a nucleus, and DNA can't leave the nucleus. And so you're going to make the transcription, you're going to make that messenger RNA in the nucleus. It will then leave the nucleus and get read by ribosomes out in the cytoplasm. In a prokaryotic cell, they have a singular circular chromosome versus linear, and they're still going to make messenger RNA, the green strand here, and it's going to get read by ribosomes to make your amino acid sequences. Nascent protein just means growing protein. And I'm like, it just is all going to happen in the cytoplasm because there's no nucleus. Now, when something happens to DNA, again, that sequence, the A's, the T's, the C's, and the G's are very specific. So if anything changes the DNA sequence, you have what's known as a mutation. Now, non-mutated DNA is known as wild type or wild strain. I always like to think it's the normal wild, DNA you find out in the wild. No changes happened. However, if an organism has some type of change in their DNA, we call it a mutant strain. There's been a mutation that has happened with that DNA. Now, there's reasons why mutations can happen. Spontaneous mutation means some type of change, some type of change in the DNA, usually because of a replication error. That when they were making new DNA, if it was supposed to bring in a T, it accidentally brought in a G or a C. And I'm like, and it happens. Induced mutations means that you've exposed the cell, whether our cell or not, we've exposed the cell to something that purposely changes DNA. Well, whether it wants to, but it can change DNA. So it's called an induced mutations. There are different chemicals, there are different factors like radiation that can change DNA. It wasn't something that just happened in the cell when it was trying to go about its business. And I'm like, it's something that induced a change in DNA. These are just some examples of chemicals. And I'm like, that can change DNA. Some radiation, different types of radiation. Yes, UV light causes mutations. And yes, changes in DNA can cause cancer. And so this is why UV light causes cancer, aka the sun, overexposure to the sun can cause cancer. It's because UV light is a type of radiation that can change mutations. And generally, mutations are not good. Now we do group mutations. There are different kinds of mutations. Now I'm going to show pictures, I like pictures, of these different mutations. Point mutations just means you're going to add, delete, or change, a base. It's at one point we're going to change just one thing. A missense mutations is a change in just one single amino acid. A nonsense changes a normal codon, a 3-letter sequence, into a stop codon. That's never good. A silent mutation alters a base, but doesn't actually change the amino acid. And then when you have something called a frame shift mutation, it's usually when you add an extra base or delete a base. That's going to change all of the codons. If all of a sudden you add one letter, it shifts everything over, hence frame shift. So this is showing some examples. So here's my normal strand of DNA. So I'm going to always compare it back to my normal strand. Here's your normal DNA sequence. Through transcription, here's your normal messenger RNA sequence. And then through translation, here's your normal polypeptide sequence. A silent mutation. I'm going to change just this letter. It was an A, I'm going to change it to a G. I've mutated it. But just one point, one single area. Now, if I go through transcription, I've got a G now instead of an A, so I'm going to bring in a C. But UUU codes for phenylalanine, and UUC codes for phenylalanine. So you still brought in the right amino acid. So although it is a change in DNA, it doesn't change the final outcome. So it's called a silent mutation. A missense mutation. So down the line, we originally had a G right here. I'm going to change it to a C. And going through transcription, I'm going to bring in a G. And when I go through and read my 3-letter codons, I'm going to bring in glycine instead of alanine. So the gly versus the ala. Now, everything else is the same. Phenylalanine is the same, the tyrosine is the same, the arginine is the same. The only thing I've done in a missense mutation, I've changed one amino acid. Well, when you have a protein sequence of 1,000 plus more amino acids long, you're like, one amino acid different, the other 999 are the same. Well, yeah, but one only amino acid change can be deadly. An example, sickle cell anemia is one amino acid different. And so it happens that just one amino acid is changed from in the normal protein strand, and it's what causes sickle cell anemia. So yeah, one amino acid change even though 999 of them are the same, can still be deadly. A nonsense mutation is, again, here's my original DNA, but I'm going to change this A to a T. And after I go through your transcription, I've now accidentally made a stop codon, which means nothing's going to happen after that. So I still brought in phenylalanine just fine, but now I made a stop codon. So I'm not going to bring in any other amino acid after that. That's going to be detrimental. Frame shift mutations, whether you insert or delete, either way, you're going to shift everything over. I'm bringing in, after these 3 A's, I'm going to bring in a T that wasn't there. So I'm going to bring in a T right here. Now, because I read everything in 3, if I bring in an extra letter, all of this shifts over. And so my 3 letters, when I read them 3 at a time, everything shifts over. Everything after a frame shift insertion, or if I just deleted this T, everything shifts over, is going to be detrimental. Is because everything's going to get read incorrectly and you're going to bring in all the incorrect amino acids. Now, it does happen that, especially with the spontaneous mutations, that cells can have mutations. And so, luckily, cells have figured out a way to repair some of those mutations. So we do have different enzymes that can try to proofread our DNA sequences. They can alert our cells that there's a mutation. Sometimes they'll just make the cell die. Like, all right, well, there's a mutation. We just need to stop the mutation where it is. So, otherwise, we do have different enzymes and different structures that can go through and fix different mutations. It can remove an incorrect base and bring in the correct base. If something was linked or bonded incorrectly, it can go through and undo that as well. So we do have some repair mechanisms for different mutations. Now, mutations generally are harmful. No one likes change. Cells don't either. Mutations are usually deadly. However, it could happen that you end up with a mutation that actually is beneficial. So organisms that have mutations that are beneficial, they are more likely to survive, they're more likely to reproduce, and this is where we start to get natural selection, that they have some trait that makes them more likely to survive. Mostly mutations are deadly and bad, but occasionally this is how we get change. And I'm like, occasionally a mutation could actually be a good mutation. Now, the last part that we're going to talk about is genetic recombination. The fact that we can move DNA around. We can recombine it with different cells. So we can move DNA around in a cell. We know what DNA does. We know how to transcribe it, translate it now, but the fact that we can move it around. Now, there are 3 major ways that DNA can move around and get recombined with other organisms. It's conjugation, transformation, and transduction. Now, bacterial conjugation is the #1 way that bacteria can share DNA with other cells. Now, they're talking. They're sharing information, just like if you would call someone up and share information with someone. Anytime they are talking when they're attached to each other, it's good for them, and anything that's good for a bacteria is bad for us. So, showing an example of how they can do this. If this bacteria wants to talk to this bacteria, they can share DNA. That's the talking part. It usually happens with their plasmid. A plasmid is just a little snippet of DNA, different from their chromosomal DNA, and it's still DNA, it's still double-stranded, but it generally doesn't code for their normal survival. That's all found in their single circular chromosome, but it's still a snippet of DNA. It's still this extra DNA. And DNA, any DNA, snippets or not, can still code for something. They can still have genes. So if these 2 cells want to attach, and I'm like, they're going to send out what's known as a conjugation pili. Now, these 2 cells don't even have to be the same kind of bacteria. They can be 2 totally different species of bacteria. But they're going to send out a conjugation pili. It tries to bring the cells as close as possible. And then one of the 2 strands, it's still double-stranded DNA, one of the strands of that DNA from that plasmid is going to go into the other cell. And it will make its opposite copy of it, so it still has double-stranded DNA. But now you have this cell that has this new DNA in it that it never had originally, and it got it from another bacteria. Now, this causes a big problem when bacteria can talk. Again, it's never good for us because a lot of times the information found on these plasmids codes for something. And one big thing we found out is that those plasmids can contain genes that code for antibiotic resistance. That there is information on this DNA that can make that bacteria resistant to different antibiotics in various methods, but it can make bacteria resistant to antibiotics. And if this bacteria is resistant to antibiotic and it just shared that information on how to do that, now this bacteria is. So if this bacteria is resistant, now this bacteria is resistant. And again, this is one really huge way on how bacteria share information and how antibiotic resistance is happening. Is you have lots of bacteria that have figured out how to be resistant, and they're all sharing their information with each other. When you pull up your PowerPoint, if you want to click on this link, it's going to show this happening. Now, another way we can get DNA from one cell to another, it's not just bacterial conjugation, but it's called transformation. Is when you're going to take a dead cell, a lysed cell. So it's broken apart, it's dead, but even though a cell is dead, it still has DNA in it. And that DNA, that released DNA, can still get inside of another cell. You just put them close together and that DNA can get uptaken into other cells. Now, it's not common. Bacterial conjugation is common. Transformation isn't common because that DNA still has to get past a cell wall and past a cell membrane to get inside of another cell. And so certain bacteria are more likely to have this happen than others. But we know that it can happen. And so if we want to get DNA, like we want to get DNA inside of another cell, we can do this in labs. It's called recombinant DNA technology. Now, Griffith is a man that did a lot of studying and figured this out. And he studied a bacteria called Streptococcus pneumoniae. Now, there's a couple different types of Streptococcus pneumoniae. And so one type is called smooth because the cells themselves have this capsule on the outside. So they look a little smooth. Now, a capsule is good for the bacteria. It provides a little camouflage so that our cells can't see it. It's harder to get rid of. It's harder to get eaten through phagocytosis. And so it's good for the bacteria. And anything good for the bacteria is bad for us. So if I took this smooth strain of Streptococcus pneumoniae and I put it into a mouse, because it has a capsule, mouse is going to die. But there's another strain of Streptococcus pneumoniae that doesn't have a capsule. So they call it a rough strain because it doesn't have that nice smooth capsule. Now, because it doesn't have a capsule, the immune system can recognize it and it can be easily eaten and gotten rid of. And so if you put this bacteria in a mouse, it's going to live because their immune system, just like ours, could get rid of it. However, so that's my little basics, 2 types of Streptococcus, smooth and rough. Smooth kills, rough doesn't. However, we're going to heat kill. We're going to kill the smooth strain. And if you kill the bacteria and you put it into a mouse, it's going to live. So we killed the bacteria. It's going to live. However, if I mixed some live rough strain with some heat-killed S strain, this allowed the mouse to live. This allowed the mouse to live. But if I mix these two together first, and then I put it in the mouse died. When you think about it, the mouse should have lived. The mouse should have lived. The mouse should have lived. However, it did not. And the main thing is what's happening in the transformation is although the smooth strain is dead, it's heat treated, we killed it, it still has DNA. And there is DNA that's going to get released when you kill a bacteria. And in that DNA, there's a gene on how to make a capsule. Because it didn't just make a capsule for no reason. There's a gene in there that codes for how to make a capsule. And that gene went from one bacteria into that live rough strain. And all of a sudden, it can make a capsule when it didn't used to know how. That living R strain, when it was still alive, it picked up the DNA on how to make a capsule. It made a capsule, and that's why the mouse died. Now again, not a lot of bacteria can easily uptake DNA because it does have to get through that cell wall and the cell membrane. However, Streptococcus bacteria of lots of different species seem to be a highly likely bacteria that can happen. It's more readily available, not available, but more readily able to happen. Now, another way, a third way that we can get bacteria from one cell into another, is called transduction, is we're going to use a virus. Now, viruses can infect bacteria. Everyone's like, what? But bacteria are bad and viruses are bad. They can't affect each other. Oh, they totally can. There are viruses that infect bacteria. Viruses just need a host cell to replicate. Bacteria is a cell. Perfect. Perfect place to get replicated in. So viruses that infect bacteria are called bacteriophages. It just confuses a lot of students. You're like, the word bacteria is in it. But it's a virus that infects bacteria. So a bacteriophage is a virus. Sometimes to simplify it, we'll just call them phage viruses and try to take the word bacteria out to be less confusing. But we're going to use a virus that infects bacteria to transfer DNA from one cell to another. Now there's two types. Generalized transduction kind of just means we're going to pick any random sequence of DNA and move it. Specializes, we're going to pick a very specific strand of DNA and move it. But here's how we're going to use that virus. So here's a bacteriophage, a virus, that's going to infect this bacteria. And when it does that, the virus injects its own viral DNA into this bacteria. Now, it's now hijacked the bacteria, and the bacteria has now become a virus-producing factory. Now, some of that viral DNA, while this bacteria is making more viruses, some of the bacterial DNA is going to mix with the viral DNA. And so some of this purple bacterial DNA is going to end up inside of a new virus. And so when this bacteria dies, this original cell always dies, when this cell dies and releases all of these new viruses, some of them contain some bacteria. And so when that virus goes and infects another bacteria, it's going to inject some bacterial DNA inside of a new bacteria. And so we've now inserted that new bacteria DNA inside of a new bacteria. We're just using viruses. So we can use this virus that infects bacteria to make lots of new viruses, but some of them will have bacterial DNA as well as viral DNA. So again, specialized means you move a specific segment of DNA. General just means kind of any random segment of DNA gets moved. Now, this doesn't always mean we go from one cell to another, but it's another way that DNA can move around. They're called transposons. They just mean jumping genes. So if this particular gene is right here on your chromosome, here's one single circular chromosome. If this particular gene wants to move, it can. It can jump from one segment to another. Sometimes it replicates in the process and inserts itself in multiple places. Sometimes as it inserts itself into multiple places, it can actually insert itself in a plasmid, which then, yes, maybe through bacterial conjugation, it can go into a new cell. But it's moving DNA from one section to another section, usually inside the cell, but sometimes if it goes to a plasmid, it can go elsewhere. Now, sometimes this can be beneficial to the bacteria if it needs it to go somewhere else. Most often, this can cause mutations when you have moving DNA, and so most often it can be harmful. Last little concept check about this. So again, we just went over lots of DNA. We went over, how they're packaged in chromosomes, genotypes and phenotypes, how DNA gets replicated, how we can then transcribe DNA into messenger RNA and translate it so that really ultimately DNA can make any protein that's necessary for the cell. And then we also talked about mutations that can happen with DNA and how we can move DNA from one cell to another or even inside of the cell. So lots of information, lots of helpful information in this PowerPoint to help complete your worksheet as well. And again, if you have any questions, please email.